Okay, so case study number 3 really focuses on pathway analysis and integrative analysis of cancer genomic data, very much in the spirits of the Cancer Genome Atlas publications. We're trying to replicate the ability to run these analyses, but in a systematic, pre-computed fashion on the web. So the case study that we're going to do is we're going to look at the question, what pathways are most frequently altered in a particular cancer type? What types of alterations, what are the most altered components of a particular pathway, and what are the relationships among these alterations? Do they tend to co-occur, or do they tend to be exclusive of one another? We're going to focus on glioblastoma. So I know that this paper's been out now for a couple of years, and everyone's probably familiar with the findings. I think the exciting thing that we're doing here is we're building systems to automatically make the observations in a very intuitive way that are being made by these fairly lengthy and exhaustive TCGA studies. And the idea is that as all of this TCGA data becomes available, copy number, expression, mutation, it'll plug in nicely into our tool set such that similar observations for other cancer types can be readily made. I'll also show you an example of things that we think that we've pulled out via our analysis that were missed by the original TCGA study. So one of the key findings in this glioblastoma paper was that there were 3 main pathways that were altered in a large fraction of the glioblastoma patients. So if you group together receptor tyrosine kinases with RAS and PI3K signaling members, this pathway was found to be altered in nearly 90% of cases. Similarly, if you group together P53 and MDM and CDKN, that pathway is frequently altered, as is the RB signaling pathway, linking together CDKs, as well as cyclins, as well as RB. So this is what we're going to show you in our tool set, but show you a dynamic view to mine this type of data. So what I'm showing you here, this is actually a beta tool, and we've actually just brought this tool down to update it, but should be back available in the near future. So I'm going to show you a workflow here in the TCGA tool. So what we're looking at here is brain cancer, and we've got brain and ovarian cancer available. And what we're summarizing are the genes and pathways that most frequently have genetic alterations in the glioblastoma TCGA study. And we've turned off gene overexpression and gene underexpression, focusing specifically on genetic alterations, namely gene mutations, amplifications, and deletions, and pathway alterations. What you can see from this analysis, and this is an analysis based on all of the raw TCGA genomic data, as well as a predefined set of pathways that we've curated here at Compendia and have used routinely in our biomarker studies. And what we're seeing is that the most standout alterations in glioblastoma are very much in line with what was published, namely the cell cycle pathway is frequently altered, receptor tyrosine kinases are frequently altered, PI3K, P10, mTOR pathway is frequently altered, as is the P53 MDM pathway. I've skipped over a couple of pathways here that are really redundant with pathways that I already spoke to. In this view here on the bottom, having selected these 4 pathways, what we're showing you is a case-by-case view. So each column is a TCGA glioblastoma case, and we're starring cases that have an alteration in the pathway. And I think what you can see here is, for the most part, glioblastomas have alterations in many, in some cases all, of these affected pathways. So that's one interesting observation, and I think poses a challenge when thinking about targeting glioblastoma. Glioblastomas often have multiple aberrations across multiple cancer pathways. So now let's drill in to some of these pathways, and we'll begin with the cell cycle pathway. So our question here is, what are the components of the cell cycle pathway that are most frequently altered in glioblastoma, and how do those components relate to one another, i.e. do they occur together or mutually in a mutually exclusive fashion? So what we're going to do is go to the pathway deconvolution tab I'm going to turn off the expression calls and focus specifically on amplifications, deletions, and mutations. On the y-axis here, we have the number of glioblastoma patients. The study includes only 91 of the cases for which we have complete genomic coverage, meaning mutation, copy number, and expression. What you can see here is that a number of alterations in the cell cycle pathway occur. We get deletions in CDKN2A and 2B. Rarely we get mutations in CDKN2A, mutations in RB1, more rarely deletions in RB1, and then also amplifications in CDK4, but in other cyclins and CDKs as well. To examine the relationships among these alterations, we simply select them. And now again, we have a patient-by-patient view. Each patient is a column here. And we're simply coding the patients for the alterations that we've selected. And what you can see very clearly from this analysis is that for the most part, patients have only one cell cycle alteration. They either have a CDKN2A, 2B locus deletion, or CDK4 amplification, or RB mutation. A couple of exceptions to that rule, but the rule holds for the most part. Also, the CDKN2A mutant cases tend not to have deletions of the locus as well. So it's mutation or deletion or amplification in one of these pathway constituents. Okay, so let's jump back to genes and pathways, and we will do the same sort of pathway deconvolution for receptor tyrosine kinases. So we know from looking at the plot that EGFR is a frequently altered receptor tyrosine kinase, and the difference between EGFR and the pathway as a whole is relatively small. So much of the receptor tyrosine kinase alteration in glioblastoma is due to EGFR. But let's see what other aberrations exist and may contribute to pathway activity. What we can see here is something that was reported in the paper, PDGRF alpha amplifications and mutations. But also, I'll point out rare amplifications and mutations in other receptor tyrosine kinases, like FGFRs and HER2 and others. So that's one aspect of an integrative analysis, is looking at the pathway level. We can also look at the gene level. So we see here that EGFR has cases that are amplified and cases that are mutated. So a question that we might have is, does amplification result in overexpression, and are the mutations occurring in the same cases that are amplified or in different cases? So I'm going to turn off the cases in gray for which we don't have mutation status. So we're only focusing on the glioblastoma cases for which we have expression, copy number, and mutation. And this data is absolutely fascinating. So when we look at expression on the y-axis and copy number on the x-axis, the first thing we see is what we would expect. And that is that cases with amplification have overexpression and actually have the highest overexpression out of all cases. There are no cases with neutral copy number that have equally high overexpression. But what's most fascinating by this data is the co-occurrence of mutations with amplification and overexpression. I might have predicted, not having looked at the data, that most of the mutations would be confined to the copy number neutral cases as an alternative mechanism to activate EGFR. But instead what we see, these cases are amplifying and overexpressing and mutating the EGFR protein, pointing to multiple selective hits on this same protein and its importance in cancer. So the last thing that we're going to look at is the co-occurrence and mutual exclusivity analysis. So now we're asking the question, for the EGFR gene amplification, are there other molecular events that occur in a mutually exclusive or co-occurrence fashion? And what we're looking at here is we're sorting by mutual exclusivity first. And what you can see here is that we're getting a statistically significant association between EGFR gene amplification and FGFR pathway amplification, deletion, or mutation, suggesting that these two events are mutually exclusive. We also see, if I sort here by p-value and focus specifically on the mutual exclusivity data, and we're going to turn off the expression data, what we see is mutual exclusivity with the p53 mutation. So not perfect mutual exclusivity, but certainly a significant trend. EGFR amplifications and p53 mutations are less likely to occur together than what you'd expect by chance. The FGFR association, you can see the odds ratio is 0, suggesting that we never have both an EGFR amplification and an FGFR pathway aberration. And similarly, we see significant trends with other aberrations, including PDGFR and RBE2F. But what we're pointing out here is that the 10 cases with FGFR pathway alteration are all wild type for EGFR amplification, suggesting that this may be a rare but alternative receptor tyrosine kinase that is a pathway that is activated in glioblastoma. I think in the paper, the EGFR and PDGFR focus was highlighted. I think what we found here is that in a smaller subset of cases, FGFRs may be an alternative receptor tyrosine kinase activated in glioblastoma. Now, the number of cases are small, and we're seeing alterations across a number of FGFRs. So this likely needs to be validated in a larger cohort. But the mutual exclusivity with EGFR suggests that perhaps these are alternative driver events in glioblastoma.